So we move into our third speaker today, who will provide the third piece of the jigsaw as to why our mental health is declining, and most importantly, what we can actually do about it. Professor David Smith is Emeritus Professor of Pharmacology at the University of Oxford. Having been head of pharmacology for 21 years, and it was during that time that his department achieved the highest rating of any pharmacology department in the world. He was also Vice Dean of the Medical School for 5 years. He is a leading expert in dementia. He helped form and head our scientific advisory board at Food for the Brain. It was his research group at Oxford that developed the scan of the medial temporal lobe that is now used internationally to diagnose Alzheimer's. He was also the first to identify homocysteine as a risk factor, a marker for Alzheimer's. His pioneering research on arresting the dementia process of brain shrinkage and cognitive decline is literally the most effective evidence-based disease-modifying treatment in existence today. Let's find out more about what really causes Alzheimer's and how to prevent it, and welcome and thank David for his excellent contribution to mental health.
So, ladies and gentlemen, going back to this slide, because of course that's a piece of broccoli. We all eat broccoli. We know it's very good for us. It's one of the green vegetables. And the Minister for Health in the UK, Matt Hancock, last November sang its praises, but he got it wrong. What he said, he was addressing a conference on preventive medicine, which is a great thing because it's good to know that the Minister for Health is supporting preventive medicine. And he gave the introductory speech in this international conference in London. And he said, for example, he said, prevention is very important, and people with B12 deficiency have a greater risk of developing dementia. But he said, no problem about that. All they have to do is to eat a lot of broccoli. Well, he was badly advised, wasn't he? So when I read this, I was just shocked. It was in the Times and Telegraph and one or two newspapers, and it reached the British Medical Journal, and so I wrote to the BMJ and corrected this awful mistake and said that it's very good to know that the Minister for Health recognises the importance of B12, but it's unfortunate that he thinks there's B12 in any plant product. There certainly is not. So I'm broadening out my title from the one that was published because I thought it was better to have a slightly wider scope, and I'm going to talk about nutrition and the prevention of cognitive impairment and Alzheimer's disease. Alzheimer's disease is the sort of end result of cognitive impairment, if you like. It's when it becomes irreversible. As Patrick told you earlier at the beginning, the number of people developing dementia or receiving a diagnosis in the UK has gone up enormously. Just 5 years ago, it was 550. In 2015, it was 790 people every day receive a diagnosis or develop dementia. That is a really alarming thought. And this paper analysed that's the actual prevalence of the disease, the incidence of the disease, but they also analysed the prevalence, as you can see from these plots. And the top one, the blue line, shows the combined prevalence of men and women with dementia as predicted from all the data they have. And as you can see, it's increasing. The prevalence of dementia is increasing, mainly, of course, because it's an age-related disease and the population is growing older. So the higher the proportion of elderly people in the population, the greater the prevalence of dementia. So the question is, can we do anything about it? So if it's age-related, then the common thought or belief is that it's an inevitable part of aging. People say that to me. They used to say that to me frequently. They'd say it somewhat less frequently now. Is that true? Is it an inevitable part of aging that if we live long enough, we'll all end up with Alzheimer's? No, it's not. The dementia is associated with specific changes in the brain. which are not found in everyone. So that's a good news. It really is a disease. It's not a natural part of aging, although it increases in prevalence with age. So here's one of the characteristic changes you find in the brain in Alzheimer's. You find deposition of neurofibrillary tangles within nerve cells in the cerebral cortex. These are nerve cells stained with a silver stain, which shows up these so-called tangles. And these are in the hippocampus, the part of the brain that is so crucial for memory. Now, the important thing is that not only does the deposition of these tangles, which is an insoluble protein called tau, phosphorylated tau, lead to malfunction of the neurons, it leads to death of the neurons. So for every one nerve cell you can stain with to show the presence of tangles, 6 nerve cells have died. So this is the main cause of the dementia is not only the strangling of the neurons, but the fact that they're dying. Now, these tangles can be studied, of course, in histopathology, and the distribution of them in the brain of a patient with Alzheimer's disease is shown here. The stronger the color, the denser the tangles. So you can see that the medial temporal lobe, which includes the hippocampus, has the highest density of these neurofibrillary tangles, but it's present in the prefrontal cortex, in the parietal cortex, and in the temporal cortex. It's not found so much in primary sensory areas. Now, that is the end result. Now, these regions of the brain that show the high density of tangles actually shrink as the disease progresses, and we'll talk about that later. The question is, how did it get there? Is it sudden? No, it's not sudden. It's a gradual process. So 5 years before the end stage of Alzheimer's, that's shown on the left there, you have what we call mild cognitive impairment, where there's a small number of, but significant number of these tangles, especially in the medial temporal lobe. Before that, there's a long period of about a quarter of a century when the tangles are only found in the medial temporal lobe. So we have a very slow development of the disease. And even before that, in young people, I'm sorry to say, you get the occasional tangle in the medial temporal lobe. So you might say, well, this is depressing. It is and it isn't. The good news about this is it takes a long time for the disease to progress, so that gives us time to intervene in the disease process. Now, the end result is shown here on the right. This is the brain of one of our patients with Alzheimer's disease, and you can see that it's severely shrunk. Most particularly, the medial temporal lobe, which contains the hippocampus, has more or less gone completely, compared with a normal, healthy brain of the same age. So, to prevent Alzheimer's disease, we have to find a way of stopping or slowing the shrinkage of the brain. That's the target. Let's try and prevent shrinkage of the brain. So we've said that one of these stories is not true, it's a myth. It's not an inevitable part of aging, Alzheimer's, but it's a true disease. The other common remark that people make to me all the time is, but anyway, doctor, it's all in the genes, isn't it? So we can't do anything about it. Well, no, that's not true either. There's only 3 very rare genetic mutations are known that cause Alzheimer's directly, that are a definite cause, and that occurs in only about 1% of all cases of Alzheimer's. So at least 99% of Alzheimer's cases are what we call sporadic Alzheimer's. They are not directly caused by mutations in a particular gene. So in that case, what causes cognitive impairment? If it's not an inevitable part of aging, if it's not in the genes, what causes it? The causes are multifactorial, and that's a very important thing to recognize. It's not one single cause. Many different factors have to come together to cause Alzheimer's disease. And the common causes that are genetic determined risk factors, that's a mutation in a gene which is common, it's called a polymorphism, in the population, but doesn't inevitably lead to Alzheimer's, it just increases the risk that you'll get it. And for those, there are many that have been discovered now, something like 70 or 80. But these are mostly a very small effect indeed, have a really tiny impact on your risk of having the disease, less than 10%. There's one called ApoE, the mutation in the gene for apolipoprotein E, which has a somewhat higher risk factor, but most of them are very, very small. So these genetically determined risk factors interact with non-genetic risk factors. And of course, the great thing about non-genetic risk factors is that they're potentially modifiable. It's not so easy to modify your genes, but you can modify the non-genetic risk factors, and that's what I'm going to talk about. So, to summarize what I've said so far, cognitive impairment and Alzheimer's disease develop slowly over many years until a threshold is reached when cognition is affected. So the question is, can we use this time window to slow down progression of the cognitive impairment and so prevent Alzheimer's disease from developing by modifying these risk factors? Here's a list of postulated non-genetic risk factors. And I say postulated because they're not proven unless you've done a trial to intervene and modify them. They're postulated, proposed, based on evidence, some of which I'm going to give you. And we could spend, well, the next 10 or 12 hours going through all of these in turn, and I don't think you want me to do that. But you see that I've highlighted some in red, and these are, of course, related to nutrition. So several of the postulated non-genetic risk factors are related to nutrition, and the question is, how good is the evidence that they are risk factors for Alzheimer's? There are three levels of evidence, if you like. One is so-called cross-sectional observational studies where you compare the presence of one of these nutritional risk factors in a population without Alzheimer's and with those levels in a population with Alzheimer's. The next level, which is better, is what so-called prospective observational studies, whereby you take a blood sample or you do a study at baseline when no one has any cognitive impairment. You follow them for 5, 10, 15, 20 years, and you see if there's any relation between the measurements you made at baseline when no one has cognitive impairment and the later development of cognitive impairment or Alzheimer's. The final level of evidence is, of course, to do a randomized clinical trial where you intervene to modify the risk factors and see whether you can actually slow down the disease process. So basically, these lines of evidence get stronger as we go down the list. There are problems with these observational studies. Most of the evidence that we have for risk factors is observational, but there are problems. Here's a good example. You could have bias or confounding or reverse causality. Here's the observation, which is a true one, made in New York in 2007, that greater intake of folate reduces the risk of developing dementia. Great. So that's the association. The trouble is, of course, it might be reverse causality, because what that means is that people with dementia will have a poor diet, or they forget to take their folic acid if they're taking supplements. That leads then, of course, to low folate intake, which apparently causes dementia. So it's what's called reverse causality. So that's why observations of this nature have to be very carefully scrutinized and checked for reverse causality. Then there's something called confounding, which is equally common, in fact, maybe more common. And here's an observation. Whole food dietary pattern protects against cognitive impairment compared with processed food consumption. So I'm sure Robert would be pleased to know that. That's an observation, very definitely confirmed. The problem is, could it be due to confounding? The way this is could be due to confounding is that it's due to people having better education who have a good dietary pattern and therefore have good cognition, or rather they have good cognition because they have better education, and that confounds the association on the horizontal line. very difficult to exclude all these things. Still, I'm going to tell you about a lot of observational studies, and most of these have been very carefully conducted to try to reduce or eliminate these problems. So what I'm going to talk about this afternoon are the following. I'm going to start talking about dietary patterns in relation to cognition. Then I shall look at some individual dietary components, and then I shall look at the interactions between micronutrients, and finally we'll end up with awareness next. So let's look at some dietary patterns. A study done in Stockholm in the Karolinska Institute compared people who were recruited at baseline with no cognitive impairment in their sort of late middle age, and followed through until some of them developed dementia, and they looked at the diets. They had two sorts of diets. One was called prudent, and the other was called a Western diet. The prudent diet is frequent vegetables, fruit and legumes, cereals, use of cooking or dressing oil, whole grains, rice and pasta, fish, low-fat dairy, and water, believe it or not. But the Western diet, as they defined, was a frequent consumption of red or processed meat, saturated and trans fats, refined grains, sugar, beer, and spirits. So I won't ask you which category you fit into, but the lunch, I think, was probably more on the left than on the right. So what did they find? They looked at cognitive decline over a 7-year period, and this is what they found. People who took a prudent diet, they declined less than those who had the Western diet, and that was statistically highly significant. So that's good news. Some dietary patterns might be beneficial. Let's look a bit more at dietary patterns. Here's a study in New York on the Mediterranean diet, where they showed that it protects against Alzheimer's by this observational study. Again, this is a study over, I think, 10 years now, where they looked at the survival of people in the cohort without Alzheimer's, and they found that more people were protected against Alzheimer's if they had strictly adhered to a Mediterranean diet than those who adhered very poorly. It was a very significant difference. So it's a dose-related effect, which is very nice, because those who adhered in a moderate sort of way, they were in between the two results. Oops. Yeah. So the Mediterranean diet in this study reduced the rate of rate of Alzheimer's diagnosis by 34%, which is a big effect. So what is the Mediterranean diet? Let's just remind ourselves of it. I'm sure you all know. I don't think I need to go through every single item there, but just have a quick look at that list. That's the classic Mediterranean diet. And the important thing, I think, is what it contains, of course, but also that it has less meat and poultry than the average ordinary Western diet. Now, in Chicago, the Rush Alzheimer's study, led by Martha Morris, has looked at a modified Mediterranean diet. Again, this is observational, and they call it the MIND diet. So the name is very appropriate, of course. And what they found with this MIND diet was an even bigger effect on slowing cognitive decline. So people who adhered very well to the MIND diet, they had a high score, declined cognitively over a 10-year period much more slowly than those who adhered very poorly to this MIND diet. So this is a modified Mediterranean diet. I'll show you in a minute what it is. They also looked at the proportion of people who are taking this diet, who developed Alzheimer's disease over a 10-year period. And again, there was a very striking and statistically significant relationship. Those who adhered strongly to the diet, in other words, had a high score, compared with those who adhered poorly, they were protected against Alzheimer's. And it's a big effect. About twice as many people who adhered well to the diet were protected against Alzheimer's as those who scored low. So what is this MIND diet? I think we need to look at that a bit more closely, because a big trial is being done in Chicago and Boston now, where they're actually intervening with this diet in 5,000 people. And that's going to be crucial, because it's only by doing a trial that we'll really know if it's a genuine result. So what is this diet? Well, we've got whole grains, green leafy vegetables equal or more than 6 times a week. So every day, basically, you need to have green leafy vegetables. Other vegetables, quite once a day. Berries, that's very interesting, at least twice a week. We'll come back to that in a minute. Fish, once or more times a week. Poultry, 2 or more times a week. Beans, 3 times a week. Nuts, interestingly, more than 5 times a week. These are portions. Alcohol, a small amount, 1 portion per day, and olive oil was in the main oil used in this by people eating this diet. Those are the positive things. The things that should be avoided in the MIND diet, red meats and red meat products should be eaten less than 4 times a week. Fast food, butter and margarine, cheese, and pastries and sweets should be avoided. So this is a very interesting diet. I like to think I'm trying to follow it, but it's quite tough. Right, so here's another way of looking at in relation to dietary patterns. Here's a study where they looked at young people, to see whether the dietary patterns when they were young predicted their cognitive scores when they were middle-aged. Now that's good, because that's less likely to be due to reverse causality. It's rather unlikely that people, middle-aged people, have actually got early dementia, and so they would be having a bad diet. So they looked at nearly 3,000 young adults up to the age of 30, and they followed them for 25 or 30 years. And again, they found that people with higher scores for the Mediterranean diet were less likely to show cognitive decline over that period. So what you eat when you're young, and I see there's a lot of young people in the audience, matters for the brain in later life. So keep that in mind. Right, so we've looked at some examples of dietary patterns. There's a lot more in the literature, but I have to move on. So let's look at the individual nutrients, dietary components, and see what we learn about that. Individual dietary components that seem to be harmful or are possibly harmful for cognition, as we've heard already today from Robert, sucrose and fructose intake may well be harmful, but there honestly aren't enough studies on that. There's a cross-sectional study which reported poorer cognition associated with the intake of sugar-sweetened beverages in adults. Several animal studies on sucrose and fructose show an impairment of cognition in animals. Another animal study showed increased deposition of the Alzheimer's amyloid and tau in the brain of sugar-fed mice. And this whole story is consistent with the fact that diabetes is a major risk factor for cognitive decline and dementia. And it's supported by recent human studies on blood glucose levels. Here's one from California, I think it is, where they looked at the relationship between measured blood glucose level, the so-called normal, in the normal range. These are not people who are thought to have diabetes or anything like that. These normal levels of blood glucose and the risk for dementia 5 years later. And they found, as you can see, that around about level of 100 was a threshold. Above that, people had an increased risk of developing dementia. So there was an 18% increased risk people who had a blood sugar of 120. That's not diabetic. So that's very worrying. Then there's a study from Australia where they looked at brain size, brain volumes, or regional brain volumes in relation to blood glucose. And again, this is high normal levels. They found decreased grey matter in the frontal cortex, shown by these red areas. Is it into focus? Well, yes, just about. These red areas had smaller volumes than people with low blood glucose. And this was in turn associated with poorer cognitive performance in these people. both in working memory, in executive function, information processing speed, and language. So several cognitive impairments with high levels of normal glucose. So the question is, should we be controlling our excessive intake of foods that increase blood glucose? The conclusion of the Australian study, I think, is worth reading carefully. These findings stress the need to reevaluate what's considered as healthy blood glucose levels and consider the role of high normal blood glucose as a risk factor for cerebral health, cognitive function, and eventually dementia. They conclude by saying, a better lifetime management of blood glucose levels may contribute to improved cerebral and cognitive health in later life and possibly protect against dementia. That's speculative, but I think a very important proposal. So those are the harmful ones. What about possibly beneficial dietary factors in relation to cognitive impairment and dementia? Here's the list that I've selected for today, but in fact the first one I won't talk about because the evidence is rather confusing, but as you probably know, it's widely thought that antioxidants are protective, but I have to say the evidence is a bit unsure. Fruit and vegetables, flavonoids, vitamin D, fish and omega-3 fatty acids, and folate and B vitamins are the ones I'm going to talk about today. So this kind of these kind of observations have been provoked by studies like this, where they looked at patterns of nutrient intake in relation to cognitive function and brain volume measurements in a big American study. So they found two nutritional patterns that were associated with more favorable cognition and with MRI showing brain volume as being maintained. And that was one that was high in B vitamins, one high in vitamin B, C, and in vitamin C, D, and E, and another pattern which was high in omega-3 fatty acids. There was a third pattern, however, in this study, which was characterized by high trans fats that was associated with less favorable cognition and with evidence of brain atrophy, regional brain atrophy. Now, in relation to flavonoids, which of course are found in many foods, we did a study in Norway a few years ago in 2,000 odd elderly people, and we looked at the dietary intake of different flavonoid-rich foods over a period of a year. The questionnaire asked them to identify their normal dietary habits over the past year, and we related this to several cognitive tests, and this shows the relationship of this food intake to semantic memory score. And you can see that eating more fruit and vegetables improved the memory of these people. Even drinking tea was improving. Chocolate, that's good, isn't it? And wine. Now, before we get too excited, let's look at some of the numbers. As far as fruit and veg is concerned, it reached a plateau, as you can see, at around 500 grams a day, which is half a kilo. That's quite a bit, so that's okay. Tea, on the other hand, reached a plateau at about, well, it didn't really reach a plateau, so people were drinking up to a litre of tea a day and apparently benefiting. Chocolate, on the other hand, unfortunately reached a plateau at around 10 grams a day, just one square of chocolate. Still, that's good. Wine reached a plateau as small as one small glass, about 125 milliliters, which is about 1.5 units according to the unit measurement. So these are statistically significant results, and what I like about them is that they're dose-related. That's very important. It's not just a one-off, shows a graded response. But we shouldn't sort of take the data as showing that we better go and drink wine. Now, in relation to fruit and vegetables, there's a particularly nice study of dietary intake in berries, which are rich in flavonoids, in relation to cognitive decline. It's part of the famous Nurses' Health Study in the US, where they studied well, altogether nearly 20,000 women, but in this case it's 16,000, where they assessed long-term dietary habits over a 20-year period. They did a cognitive assessment at the age of 74. And they found that greater intakes of blueberries and strawberries were associated with a slower rates of cognitive decline in 5 of 6 different tests, cognitive tests. So this was a pretty robust result. Women who ate 2 or more servings of berries per week delayed cognitive aging by an equivalent of 2.5 years. That's a really striking effect. I think it's a very important study. And what's interesting is these berries figure in the MIND diet, which I talked about earlier from Chicago. So it seems as if berries are good for you. What about tea? Well, there's a study from Singapore longitudinal aging study, where they looked at nearly 1,000 community-dwelling people aged 65 at baseline, and they followed them for 5 years, and 72 became cognitively impaired. They found that the risk of cognitive impairment was higher in those who drank no tea at all. Surprisingly, people drink no tea. Anyway, apparently they did. 297 drank no tea. Those who drank black tea were partly protected, and those who drank green tea were protected even more. So tea seems to be good for you. Now, the chocolate is a very interesting study, because we found 10 grams a day was rather good. And here's a trial where they gave cocoa-enriched drinks to people, and at 3 different levels, a small amount, an intermediate amount, and a high amount, and they assessed their cognition after an 8-week period. So this is a very short trial. It's not actually a trial on dementia or anything. It's just a one-off clinical trial. But it's a dose-related effect. They also found in those who had the highest intake of cocoa improvements in cognition generally, that's insulin resistance and various blood pressure and other parameters, metabolic disease. So this is good news. Then you read the small print. The study was supported by Mars. Well, it doesn't invalidate it, but as we heard earlier, there's strong commercial interest in all these issues. Let's look again at the alcohol story. So I told you that in our Norwegian study, we found that 1 small glass, about 125 milliliters of wine, seemed to improve your semantic memory score. But there's been an absolutely huge study in London, the so-called Whitehall study, Whitehall 2, where they took several thousand civil servants and followed them for many, many years, I think 30 years almost, and they looked at their diet and all sorts of factors. And here's the study of 9,000 of these people, of whom 397 developed dementia over a 20-odd-year period, 23-year period. And this is the hazard ratio for dementia in these people. And you can see that people who abstained from alcohol actually had an increased risk of developing dementia, a 47% increased risk. Significant, not huge, but still worrying. But then it decreased the risk. So by the time they were taking sort of 5 or 6 units a week, their risk was not significant. But then as they took more and more alcohol, the risk increased. So if they took more than 14 units a week, interestingly, that's what the government has recently recommended as a limit. We shouldn't be taking more than 14. Then the risk of developing dementia began to increase. So this is a really nice study on a large population. Let's turn now to another micronutrient. Well, not really micronutrients, because of course we get our vitamin D from exposure to sunshine, but it is present in some foods. Anyway, low levels of vitamin D are definitely a risk factor for developing dementia, and depending on the level, you have a greater risk of developing dementia. So those who are just deficient have a slightly increased risk. Those who are severely deficient have a very considerably increased risk. So the numbers here are quite alarming, I think. So the deficiency increased the risk of Alzheimer's disease by 69%. 42% of white American adults are deficient in vitamin D. 82% of black Americans are deficient, because of course the pigment reduces the effect of the UV light on the skin. And 60 to 80% of middle-aged British men were found to be deficient in vitamin D. Even in Australia, or sunny Australia, 31% of Australian adults are deficient in vitamin D. So I think there's a really urgent need for a clinical trial of vitamin D in the prevention of cognitive impairment of dementia. The high prevalence of the deficiency is quite alarming. I said one of the factors I'd look at were omega-3 and fatty acid and fish. And here's, again, our Norwegian study where we looked at fish intake in this population of over 2,000 Norwegian elderly, and this time in relation to episodic memory score. And you can see that a nice dose-response relationship. The usual intake of fish, the higher the intake, the better the memory score. Again, there was a plateau around 80 grams a day. Quite a lot, really. So above 80 grams a day, it didn't seem to improve things, but there was a dose-related response. There's been a meta-analysis on this whole issue by Beidoun, National Institutes of Aging, and she found that eating fish once or more a week reduces the risk of Alzheimer's disease by 33%. That's compared with those who eat fish less than once a week. So that's, again, a significant effect. Now, fish, as we've heard from Michael, is a rich source of several micronutrients, not only omega-3 fatty acids, such as DHA, but vitamin B12 and selenium. So we don't know which of these micronutrients it might be, or if it's all of them. So let's look at DHA. Plasma DHA has been measured in the Framingham Heart Study, nearly 900 people, in relation to the later development of dementia after a period of 12 years. And you can see that the cumulative incidence of dementia is much reduced in those in the top quartile of DHA levels in the plasma. So DHA protects by 47% those from developing dementia. Again, a meta-analysis by Beidoun shows, on the whole, a striking protective effect of omega-3 fatty acids and fish intake on the development of the risk of developing dementia. So the pooled relative risk of Alzheimer's disease was 0.67, in other words, 33% protection against Alzheimer's in those with the high levels of omega-3. Now, in this kind of statistical analysis, you can estimate something called the population attributable risk of a disease. And this is very interesting. This is the proportion of, in this case, Alzheimer's that can be possibly caused by low levels of omega-3. So she worked out the population attributable risk of Alzheimer's for low omega-3 fatty acid levels in the intake or levels is 22%. So 22% of people with dementia might be caused by low omega-3. Finally, my own field, look at the B vitamins, folate and B, and the other B vitamins. This, again, we have to have a little bit of biochemistry, I'm afraid. The B vitamins are crucial for the metabolism of the amino acid homocysteine, which is central in the methionine cycle. And you can see that 3 B vitamins, folate, B12, and B6, are crucial for the operation of this cycle. So the levels of these vitamins can regulate the level of homocysteine in the body, and that leaks out into the blood. So if you measure blood homocysteine, it's a good marker, if you like, of the B vitamin status. So again, in the Framingham study, they measured homocysteine at baseline when no one had any sign of cognitive impairment. They followed them for 8 years, and 111 developed dementia, and this is what they found, that people with the high levels, tertile 3 of homocysteine, which we abbreviate THCY, had a greater risk, nearly twofold risk of developing dementia compared with those in the lower levels of homocysteine. Again, a meta-analysis has been done, and the ratio is almost doubled. So people with high homocysteine have an almost doubling of the risk of developing dementia and Alzheimer's. This is Alzheimer's disease, actually. So again, we can do the population attributable risk calculation, and again, it turned out by chance to be the same as omega-3. About 22% of dementia may be due to raised homocysteine. Now, these are big numbers. These are millions and millions of people every year around the world, if they're really true. So, let's summarize so far about omega-3 and B vitamins. Low fish intake or low blood levels of long-chain omega-3 fatty acids are risk factors for Alzheimer's disease. Low folate and B12 status, as indicated by raised plasma homocysteine, are risk factors for cognitive decline and dementia. And each of these risk factors has, as I've said, a population attributable risk of about 20%. So the question is, what is the effect of modifying these risk factors on cognitive impairment or even on dementia? Well, there's been no good studies on dementia. Well, I'll come on to one in a minute or at the end. But on cognitive decline, there have been clinical trials. So you can lower homocysteine by giving B vitamins, and two trials, the FACIT trial in the Netherlands on folic acid alone, and our VitaGOG trial from Oxford, where we used folic acid B12 and B6, showed that lowering homocysteine by giving B vitamins did indeed slow cognitive decline. Eating fish or supplementing diet with omega-3 fatty acids can slow cognitive decline. I say can because it doesn't always. In fact, for both these risk factors, trial results have not been consistent. There have been many negative trials, and that somewhat leads people to be skeptical. But why are they negative? Why is there disagreement? I'm going to show you the reason. And the reason we discovered when we started measuring the rate of shrinkage of the brain, so I have to sort of have an intercept on brain shrinkage. As we age over 60, the brain is shrinking at a rate of about 0.5% per year. So there's not many in the audience of that age, I think. But you're losing tissue at the rate of 7 milliliters per year about. It's a bit worrying. Now, the trouble is it gets worse. Was those with memory problems, or if we define it as mild cognitive impairment, which is a clinical syndrome you can diagnose, show a faster rate of shrinkage of about 1% per year. Patients with Alzheimer's, it's much faster. It's about 3% per year. So the question is this a gradation? Is this a progression of brain atrophy as cognitive impairment and dementia develops? Well, many risk factors for Alzheimer's disease have been shown to be associated with an increased rate of brain atrophy. Quite a big list. Smoking, diabetes, low omega-3, physical inactivity, low adherence to Mediterranean diet, high blood pressure, atrial fibrillation, high homocysteine and low B vitamins. So this is clearly, brain atrophy is a useful marker, a biological marker for what's going on in the brain. Now, we looked at B12 in relation to brain atrophy in two studies. The top one was done in Oxford on 200-odd normal community-dwelling elderly in Oxford, and the bottom one was done on 2,000 normal community-dwelling people in Stockholm. And we measured B12 and we measured the rate of brain atrophy or shrinkage over several years. And here's the Oxford results. This is the percent change in brain volume over a 5-year period, plotted against, on the horizontal axis, the vitamin B12 level at baseline. So that's the level when they came into the study. Wow, you can see something rather alarming. You can see that the lower the B12, the faster the brain is shrinking. The levels here are not deficient. None of these people were deficient. The deficiency cutoff is traditionally considered to be 148. So these are people in the normal range of B12 whose brain shrinks the lower the level. Have I just said there's something in the blood that may determine the rate at which your brain is shrinking? I have. But you could say, oh, it's just a coincidence. So we did a trial. We did the Vitagog trial, which recruited people with mild cognitive impairment who have a rate of brain shrinkage of about 1% per year. And we randomized them, and we gave half of them placebo, and the other half we gave a mixture of folic acid, B12, half a milligram, and B6. And we followed them for 2 years, and we measured the rate of brain atrophy, and we measured various cognitive measures. And as you can see from the title of this paper, we did manage to show prevention of grey matter atrophy in people with mild cognitive impairment by giving B vitamins. So, in those with high homocysteine at baseline, in other words, in those with low B vitamin status, as indicated by the high homocysteine, they responded to the B vitamin treatment with a marked slowing of the rate of brain atrophy, slowed by 53%. When we looked at specific brain regions, as I'll show you in a minute, we found that the slowing of atrophy of these special brain regions which go in Alzheimer's was even stronger. It was 90% slowing. And finally, this treatment slowed, or even in some cases completely stopped, cognitive decline. So memory decline was actually stopped by this treatment. So here's a slide showing in yellow, the areas in the brain of the placebo group which showed significant atrophy, highly significant, as you can see, over the 2-year period. And I've indicated the medial temporal lobe with the red arrow. That's where the hippocampus is. And you can see that that's one of the areas that shows marked atrophy over the 2-year period. So that's in the placebo group. So what happened in those where we treated with B vitamins? That's what happened. I think you'll agree, a pretty dramatic reduction in the volume and in the extent of the atrophy of these brain regions, including particularly the medial temporal lobe, which is much less atrophied. So this shows that lowering homocysteine with B vitamins can actually modify the disease process. So we did some statistics to look at the causal pathway. Oh, sorry, I can't resist showing you this nice picture that my imaging colleague produced. The areas in blue are those areas protected by B vitamins. So it's not the whole brain that's protected, it's very specific areas. And these specific areas are, of course, important in cognition. So we did some statistical analysis as to what the causal pathway is, and you could say that we have a kind of matrix here with Vitagog. We have B vitamin treatment, we measured brain shrinkage, we measured plasma homocysteine, and we looked at cognitive decline. And what we did a Bayesian acyclic graph analysis, which is a way of looking at mediating pathways. And we found that, in fact, B vitamin treatment, in particular the B12, turned out to be important, lowers plasma homocysteine, which in turn slows brain shrinkage, which in turn slowed cognitive decline. So this is a causal pathway. These arrows indicate causal links. So if we summarize then the outcomes of this trial, the B vitamin treatment slowed whole brain atrophy in mild cognitive impairment. It slowed atrophy, especially in those brain regions affected in Alzheimer's, by as much as 9-fold. Slowed cognitive decline in several domains and improved clinical status. I haven't shown you that, but it actually improved the clinical status. They got better. So overall, we think the B vitamins had a disease-modifying effect. Now, I can't resist saying this, that the drug companies have been spending, the estimate is about $200 billion so far on trying to find a disease-modifying treatment for Alzheimer's disease. They've completely failed. Every treatment, every trial they've ever done has failed on the beta-amyloid hypothesis. Here's a simple nutritional intervention that slows the disease process. Certain corollaries, though. These responses in people with cognitive impairment only occurred in subjects with baseline homocysteine above the median. In other words, they had impaired B vitamin status, but they were not deficient. And as I will now show, and finally end, those with good omega-3 fatty acid status. And I'll start by reminding you that what I said earlier, that omega-3 fatty acids can affect the rate of brain atrophy. Here's a very nice study where they measured the red blood cell levels of omega-3 fatty acids, and they found that the higher the level of omega-3 in the red cells was associated with, the lower the levels of omega-3 in the blood cells associated with smaller brain volume, greater white matter damage, and poorer cognition. This is in the Framingham cohort. None of these people had dementia. So this is before dementia. So with that finding, we had in the VitaCog, we had 90 people or 80 people where they took placebo, and we measured the rate of brain atrophy over 2 years. So we had some nice data there, brain atrophy over 2 years in normal people. So we wanted to know whether does baseline omega-3 status influence the rate of brain atrophy in the placebo group. From that previous paper, we sort of suspected that it might do. The second question that we asked was, does baseline omega-3 level, influence the atrophy and the cognitive responses to B vitamins? In other words, is there an interaction? So the first question, does omega-3 level measured at baseline influence the rate of brain atrophy over time? And you can see in this plot the level of omega-3 fatty acids, this is plasma level of total omega-3 fatty acids, had no effect on the rate of brain atrophy, which was 1% a year in all these people, independent of the omega-3 level. But these people in the blue, their homocysteine was high. Well, high, it was above the median. What happened in those people where the homocysteine was low? Was lowered by giving the B vitamins. You can see a dramatic difference. That I've missed out a slide here, I'm sorry. Anyway, what we found was that people with low homocysteine, in other words, good B vitamin status, were protected by omega-3 fatty acids from brain atrophy. And this is, this is the slide. I do apologize, I'm getting cross-eyed. So these people in red had homocysteine below 11, and they were protected by high levels of omega-3 at baseline. So omega-3 can protect the brain only in those with low homocysteine, in other words, with good B vitamin status. So that's the first question, the answer of which was yes, but only in those with low homocysteine. The second question, does baseline omega-3 status influence the slowing of atrophy and of cognitive decline in response to B vitamins? And I won't show the data, but the answer was yes. Only those people with high omega-3 at baseline showed slowing of brain atrophy and slowing of cognitive decline in response to B vitamins. So here's the conclusions from VitaCog. Omega-3 fatty acids only appear to protect the brain in people with low homocysteine, in other words, good B vitamin status. B vitamins only appear to protect the brain in people with good omega-3 fatty acid status. So these, frankly, unexpected interactions could explain why some omega-3 fatty acid trials have failed, and they could explain why some B vitamin trials have failed, because the people didn't have the right composition of the other micronutrients. They illustrate the importance of interactions in nutrition, and hence the key role of dietary patterns, which I talked about earlier. So I conclude with some very new results which have just come out online. This is a famous omega-3 trial in Alzheimer's disease done in Uppsala in 2006. It was the first trial where omega-3 fatty acids were given to people with Alzheimer's disease in the belief that that might slow down cognitive decline. But unfortunately, they found no beneficial effect on cognition of supplementation with 2.3 grams per day of omega-3 fatty acids, DHA and EPA, and apparently a failed trial. So this has sort of gone down in the literature as a failed trial. Omega-3 fatty acids are no good in treating Alzheimer's. But in view of our results from VitaCog, we thought, maybe we should look at the homocysteine levels in these people to see whether that influenced the response of these people to omega-3 treatment, and it did. We found that in just those patients with low homocysteine in this trial from Uppsala, omega-3 treatment did slow cognitive decline, and therefore there's evidence of interaction, I think, with B vitamin status. So omega-3 fatty acids need to interact with B vitamins in order to benefit the brain. That's our conclusion from this trial. Now, this whole business of prevention, I hope for you, and that's why you're here, it's sort of self-evident that it's important and we should be doing it, but it certainly wasn't self-evident in the Alzheimer's field a few years ago. And in 2013, the British government invited the health ministers from G8 countries, Russia was involved then, to a conference on dementia. And I was curious to know what the agenda was going to be, so I contacted the Department of Health, and after a lot of hard work, was very difficult to get through, civil servant who was organizing the meeting, and I said to him, have you got a session on prevention? Prevention, he said? You can't prevent dementia? So I said, oh my God. So I said, could I come and see him and talk about it? And eventually Helge Ressum and I, visiting professor from Oslo, who's a nutritionist, we made an appointment with this chap, and he said, I've really only got 20 minutes, I'm so busy. An hour and a half later, he said, well, he rather convinced me that perhaps prevention should be on the agenda. So I'm glad to say it was on the agenda. But we got so annoyed by their attitude, the Department of Health, that we circulated an email petition to a lot of scientists around the world, 111 in fact. This was Christine Yaffe from San Francisco and myself. And we found 111 scientists from 36 countries who signed this statement, and this was published on the day of the meeting. And this statement was sent to all the health ministers of the G8 countries. And the statement called upon governments to make prevention of dementia one of their major health aims. We estimated in this document that about half of Alzheimer's disease is caused by known risk factors, and that taking immediate action might prevent about 1/5 of predicted cases by 2025. But that was immediate action in 2013. Well, it was partial success. The last 40 minutes of the Dementia Day was on prevention. And the great thing was that the declaration that came out from the conference, from the summit, made these statements. The first one is extremely important because that's the public perception as I started. We recognize that dementia is not a normal part of aging. And then in the communique from the summit, they made this statement. History shows major diseases can be made manageable, even preventable, with sufficient political will. Rather key statement. We recognize the importance of taking a comprehensive and coordinated approach to the prevention of dementia, tailored to national and local needs, and to take preventive measures in the near term based on existing knowledge. Well, they recognize it, but they haven't done a damn thing, frankly. Anyway, at least this is the official report, so it's good news. So let's look at the cumulative possible impact of prevention. I've listed here the population attributable risk to some of these risk factors that I've been talking about today to give you an idea of how important they are in causation of dementia. So high blood pressure is probably causing about 10% of dementia, high cholesterol about 14%. Physical inactivity, one of the most important causes, seems to cause as much as 32%. Smoking, major cause, 31%. Low education, sadly, is 24%. As I've already told you, high homocysteine and low fish intake and omega-3s each cause about 22% of dementia. Now, of course, you must not add these up because Many of these risk factors are found in several of these risk factors are found in the same person. But this gives you the idea of the scale of what we can do in prevention. If we can prevent 25, 30, or 50% of dementia, that would have a huge impact in the world. Huge. So what should we do about it? Well, first of all, we've got to improve our lifestyle, and I can't resist showing you this report that came out just recently in Lancet Neurology from the global study on Alzheimer's disease and dementia, systematic study of the global burden of disease studies. What they concluded was that 28.8 million disability-adjusted life years, which is the measure they use, were attributed to dementia in the world in 2016, of which 6.4 million, 22% of these, could be attributed to the modifiable risk factors that they define in the global burden of disease study. That's not the same as we define, not all of them. And those risk factors they identified as high BMI, high fasting plasma glucose, smoking, and a high intake of sugar-sweetened beverages. So, Robert, you should be pleased with that. Maybe you wrote it. So that's what the global burden of disease study suggests. Again, prevention is feasible. So what do you do about it yourselves? Well, first of all, you should tell everyone that we can prevent Alzheimer's disease, especially if we start in midlife. Tell your government to allocate more money to prevention. Only about 3% of the health expenditure of OECD countries is spent on prevention of dementia. It's tiny. But dementia costs a country more than the heart disease, stroke, and cancer combined. You heard that from Patrick, I think. And finally, practice prevention on yourself. Improve your lifestyle and follow a few key steps. This is Smith's suggestion. Don't sort of say it's the Bible, but my suggestion is you practice prevention yourself. Start young, age 40 to 50. First of all, stop smoking. Exercise at least 30 minutes walk every day. Take your high blood pressure pills and your diabetes pills, don't forget them. Increase the Mediterranean elements in your diet. Eat your 5 a day, fruit and veg, and include some berries. Eat fish once or more a week. Watch your blood glucose. Make sure your vitamin D and B12 status are good. If you do develop memory problems, check your homocysteine, and if it's high, take B vitamins and omega-3 fatty acids, but you should consult your GP. Keep active mentally and socially. So what I've tried to show today, ladies and gentlemen, is that nutrition is important. Nutritional intervention is a valid approach to the prevention of cognitive decline and dementia and could have a major impact on disease incidence. Combinations of different nutrients are likely to be needed, which might explain why dietary patterns are so important in prevention. Thank you very much. And good luck.